So in this lecture, what we will do is we'll talk about bioenergetics. In other words, we're going to talk about the energy, the involvement of energy within molecules in metabolism, in metabolic reactions. So let's go on. So every molecule has a potential energy, and we already talked about that before. And we said that these energies exist within bonds. Now, this energy is known as the Gibbs free energy, or G. So basically what G is the maximum amount of useful energy that can be obtained from a reaction, and that is called delta G, which is basically the G of products minus the G of reactants, and that tells us the change in free energy between products and substrates. So you can have um, a situation whereby the G of products is higher than G of reactants, okay? And this is known as inorganic reaction because energy must be applied. It has to be added into the system, and that's what's called inorganic, okay? So it goes into the system. Now, so energy has to be added. It has to be absorbed by the system. Now, this reaction is not really spontaneous. That is, spontaneous in a sense that it means that it's not favorable, okay? And delta G is higher than zero. It's more than zero. So G of products can be, let's say, something like +5. G of reactants can be something like +2 kilocalories per mole, meaning that you have to add 3 extra kilocalories per mole in order to convert reactants to products. Or a reaction can be exergonic, meaning that the energy would be released out of the reaction. Okay. In this case, we say that reaction is spontaneous. In other words, it is a favorable reaction because it means that reactants have enough energy to convert themselves, to convert themselves into products, and the extra energy would be released. And usually it is released in the form of heat, but it can be converted into something else, like formation of other molecules like ATP, for example. Now, the thing is here is that now the value of delta G, not prime, okay, tells us something. It tells us a few things actually. It tells us whether the reaction requires or releases energy. That's one. It tells us what the amount of energy involved in that reaction. It tells us what the ratio of products to substrates is. It tells us if it is a favorable reaction to the cells or not. So if delta G is less than 0, we say that the reaction proceeds towards formation of products. Okay, so it's favorable that reactants would be converted to products. If delta G is more than 0, it means that the reaction would proceed in the opposite direction, that is, from products to reactants, because products have more energy. Now, in terms of enzymatic reactions, the same thing applies. Okay, so in enzymatic reactions, we have substrates and we have products. And similarly, enzymatic reactions can be exergonic and they can be endergonic as well. Okay, so except that for enzymatic reactions, now pay attention to this. Now, enzymes do not reduce the delta G of reactions at all. So notice that reactants have the same total amount of G within their bonds. Same thing with products. So this is the uncatalyzed reaction without any enzyme, and this is the catalyzed reaction in the presence of enzyme. So notice that the G's of reactants and products do not change. Whether we're talking about an exergonic reaction or an endergonic reaction, same thing. What enzymes do is that they reduce the activation energy. okay, the energy that is needed to push the uh, reaction forward. So that's one. Now, the other thing that is important to mention is that if I say that a reaction is spontaneous, now it doesn't really mean that it is spontaneous, that it doesn't mean that it happens by itself. It just means that, so delta G naught prime is not really an indicator of the velocity of the reaction, if it is fast or slow. It doesn't tell us about the rate at which equilibrium can be reached. It doesn't tell us something about how long a reaction would take place, the time that it needs for it to happen. It doesn't tell us that. Now, because these things, that is the velocity of the reaction, the time it takes for it to take place, depend mainly on the efficiency of the enzyme as well as how much enzyme there is in the system. This diagram to the right represents the metabolic pathways that take place in cells. So you can see how complex it is and you can see how intricate it is. Now, right here, this is the glycolytic pathway, glycolysis. This is the citric acid cycle or Krebs cycle. So these two pathways lie in the heart of all metabolic pathways, and it's almost like connected to almost everything. So metabolic pathways can be divided into two types. You can have anabolic pathways. These are usually endergonic reactions. Well, anabolic pathways build up molecules. So you start with smaller molecules, small subunits, and they get connected to each other, forming larger molecules. So that requires energy. It's like, if you build a house, energy is needed to build that house. Now, on the other hand, you have catabolic reactions, catabolism. Now, these pathways usually they are exergonic. That is, energy is released. So it's like, when you play sports, if you run, jog, play basketball, whatever, energy is produced. Okay, energy is needed and energy is produced. You metabolize, you break down glucose, for example, to produce energy. So usually these reactions are exergonic. Now the thing is that metabolism is linked via a series of chemical reactions, okay, all of these pathways, and you will learn about most, if not all, of these pathways in this course and beyond. Now remember something, the first law of thermodynamics tells us that energy is conserved. It doesn't get lost. It's there, it exists. So if energy is released, it can be released in the form of heat, for example. Okay, energy can be used as well in the form of heat or biochemical energy, energy in bonds to build up larger molecules as well. So in one form or another, energy exists and it is preserved and conserved. Now, in a previous slide, I talked about delta G naught prime. But there are different definitions or different terms for G or delta G. Now we have the delta G naught prime. This delta G represents the amount of energy that exists in a reaction, products and reactants, whenever you have 1 molar concentration of reactants and products at standard conditions. And the standard conditions are 25 degrees Celsius and 1 atmospheric pressure, as well as pH of 7, neutral pH. Now this is delta G naught prime. We also have delta G naught, okay, and that represents what is known as standard free energy difference of a system, that is difference between G of products and G of reactants, whenever you have 1 molar concentration of reactants and products, but not necessarily under standard conditions, that is 25 degrees Celsius, 1 atmospheric pressure and pH of 7. And then you have delta G. Now this is the actual delta G of a reaction. That's the delta G that exists, or the delta G of a system at any condition. All right. Now remember that the value of delta G not prime tells us if the reaction requires or if it releases energy. It tells us what the amount of energy that is involved, and it tells us something about the ratio of products to substrates at equilibrium. So what do we mean by equilibrium? Now, chemical equilibrium, it means that it is a state where the rate of the forward reaction and the reverse reaction, that is the state whereby the concentrations of reactants and products are not changing. Okay. Now, it doesn't mean that this delta G is zero. Okay, so at equilibrium, it doesn't mean that delta G would be equal to zero. Now, when delta G is equal to zero, it means that the reaction is at equilibrium, meaning that the quantities of reactants and products do not change relative to delta G not prime. Because delta G is equal to delta G naught prime plus RT, that is pressure times temperature times the natural log of concentration of products over concentration of reactants. And this is true for this reaction. A plus B forms C plus D. So you have the reactants, A plus B. The products or the substrates are A and B. The products are C and D. And delta G naught prime, this is the at standard conditions, whenever you have 1 molar concentration of reactants and products, is equal to minus the pressure, which is constant, times the temperature of the system, times natural log of the equilibrium constant, which is equal to concentration of products over concentration of reactants. Okay, so whenever this value right here is equal to this value right here, it means that delta G would be equal to 0. Now, pay attention to the charge as well, and we will talk about the charge right now, how it changes. So let's look at this problem right here. The delta G naught prime for the conversion of glucose 6-phosphate to glucose 1-phosphate is plus 1.65 kilocalories per mole. What does that mean? It means that this reaction, converting glucose 6-phosphate to glucose 1-phosphate, is an inorganic reaction. The question is, what is the ratio of glucose 1-phosphate, the product, to glucose 6-phosphate, that is the substrate, at equilibrium? So we want to calculate this ratio right here. Products, concentration of products over concentration of reactants, okay? Or this equilibrium constant right here. Now, we know that delta G naught prime is equal to minus RT times natural log of the equilibrium constant. So for this reaction right here, that's what we want. So the constant R, which is pressure, is 1.98 times 10 to the minus 3. Now, you don't have to memorize this. I'm going to provide you with these numbers if I bring you a problem like that in the exam. And the temperature is in Kelvin, by the way. It is not in Celsius. Now, 0 degrees Celsius is equal to 273 Kelvin. So 25 degrees Celsius is basically 273 plus 25 Kelvin. So RT, this value right here, is equal to the sum of this, times the amount of pressure, which is, again, it's constant. Now, and this would be equal to 0.593 kilocalories per mole. Now, so we have this right here, minus RT. We have this RT. Now, this means that delta G naught prime, which is plus 1.65, is equal to minus 0.593 times the natural log of the equilibrium constant, or the ratio of product to substrate. And this means that the natural log of product over to substrate is equal to minus 2.78. And then the ratio can be calculated. You can use a calculator, and it would be equal to 0.062. So the ratio is basically of glucose 1-phosphate to glucose 6-phosphate is equal to, at equilibrium, is equal to 0.062. Okay, so we know that the delta G naught prime for this reaction, glucose 6-phosphate for this reaction, glucose 6-phosphate converted to glucose 1-phosphate is plus 1.65. So my question for you is, what is the delta G naught prime for the reverse reaction? That is glucose 1-phosphate to glucose 6-phosphate. And the answer is, it's going to be minus 1.65. So all you need to do is really change the sign right here from plus to minus, from positive to negative. So if a reaction is endergonic, the reverse reaction is exergonic. Okay, same value, different sign or opposite sign. So how is delta G of a reaction calculated? Now we're not talking about delta G naught prime. We're talking about the delta G of a reaction at any condition or in any system. Now this delta G is influenced by a number of factors. And these are the initial concentrations of substrates and products, the temperature of the system, the pH, and the pressure. Now we're going to keep pH the same, okay? We're not going to change it. It's going to be 7, okay? So don't worry about it. Now, so we want to calculate this, okay? And it would be equal to delta G naught prime plus the pressure times temperature of the system times the natural log of product concentrations of products over concentration of reactants. Now, here's some math. If the equilibrium constant is more than 1, meaning that you have more products than reactants, okay, this means that the natural log of this value would be more than 0. So that's math. Now, on the other hand, if you have more substrates than products, it means that this value right here, this ratio right here, would be less than 1. And the natural log of something that is less than 1 would be less than 0, meaning that this positive sign right here would become negative. And delta G in this case would be less than delta G naught prime. Okay, so if you have the equilibrium constant more than 1, this value right here would be more than 0, meaning that delta G would increase, would be higher than delta G naught prime. But if you have more substrates than products, then this value would be negative, okay? And delta G would be delta G naught prime minus whatever value you have here, so delta G would be lower than delta G naught prime. So here's a simple problem right here. Consider this reaction, A plus B, giving us C plus D. Now, delta G naught prime of this reaction is plus 0.4 kilojoules over per mole. Okay? So it is an endergonic reaction. Now, what if you have 1 molar A, 1 molar B, 0.1 molar C, 0.1 molar D, and these are all added to a container at room temperature? Which of the following statements is true? Now, think about it. So you have more substrates than products. So delta G would be less than delta G naught prime. So you can really predict that the reaction would become exergonic. So here you have the reaction will proceed in the forward direction to reach equilibrium, or the reaction will proceed in the backward direction to reach equilibrium. That is, it would A, meaning that the reaction would proceed in this way, B, meaning that the reaction would proceed in the opposite way. The reaction will not proceed in either direction. It's already at equilibrium. It is not. It is not at equilibrium. Okay, and it cannot be determined. Okay, what happens is that actually the system or the reaction would proceed in the forward direction. Okay, it would go in this direction because you have more substrates than products, so delta G would be lower than delta G not prime. Here's something extra, and that is, when we say that a molecule has a specific free energy, what does that really mean, that it has specific free energy? What does it mean that a reaction has a change of free energy between products and reactants? What does G really mean? Well, G actually is equal to the following: H minus T times S. Now, H is enthalpy. Enthalpy means the amount of energy within bonds of molecules. So whenever we want to calculate the amount of energy, the potential energy, the free energy within a molecule, it's really equal to enthalpy, which is, or the amount of energy, amount of heat within bonds of a molecule. T is the temperature of the system that that the molecule exists in, and it is in Kelvin. And remember, 0 degrees Celsius is equal to 273 Kelvin. And S is entropy. Entropy means disorder of a system. So whenever the system is disordered, okay, it means that G becomes lower. Okay, so whenever the system is disordered, it's not organized, the amount of energy that it has is lower than if it is ordered. So basically, whenever you have water as ice, it has less entropy than water when it is in liquid form. Okay, so the G of water as ice would be higher than G of water in the liquid form. So for any reaction, delta G would be equal to delta H, the change of enthalpy between products and reactants, minus the temperature of the system times the difference of entropy between products and reactants. So whenever delta H is more than 1, the reaction is considered to be endothermic, meaning that it absorbs energy. And whenever delta H is less than 1, it means that the reaction is exothermic. Now, the thing is with entropy is that it follows the second law of thermodynamics that tells us that the universe tends toward disorder. So if it is left alone, it tends to go disordered, meaning that energy is required to contain it and to keep it in an ordered form. Here's something important about delta G's of sequential reactions, and that is they are additives. What does that mean? It means that let's say that you have two reactions, glucose to glucose 6-phosphate, and you have a second reaction, glucose 6-phosphate to glucose 1-phosphate. The question is, and this is the question, what is the delta G of the reaction going from glucose to glucose 6-phosphate? Well, here you have two reactions. You have the first reaction, glucose to glucose 6-phosphate. And you have a second reaction, glucose 6-phosphate to glucose 1-phosphate. Now, the reaction glucose 6-phosphate to glucose 1-phosphate, delta G, is plus 1.65 kilocalories per mole. And the first reaction, that is glucose plus ATP, giving us glucose 6-phosphate plus ADP, the delta G, not prime, is equal to minus 4 kilocalories per mole. So therefore, the whole reaction, so delta G of the whole reaction that is going from glucose to glucose 1-phosphate is basically delta G not prime of the first reaction plus delta G not prime of the second reaction. Okay, that is, it is plus 1.65 plus minus 4, and that would be equal to minus 2.35. So meaning that this whole reaction is actually exergonic and it is favorable. Okay, so let's go into the second part of this lecture, and that is, how do cells make an unfavorable endergonic reaction exergonic or favorable? How can cells make a non-spontaneous reaction a spontaneous reaction? Now, the first strategy is using ATP in coupling reactions, okay, or coupling ATP to an unfavorable reaction. Now, ATP is a high energy molecule. Within its bonds, specifically the phosphoanhydride bonds, these bonds right here between the phosphates, there is a lot of energy, and this energy can be released. Now remember that it can be released in the form of heat, but cells convert this energy into biochemical energy, okay, changing molecules, changing structures of molecules. Okay, now why is it that ATP has a lot of energy? Why is it that it's favorable for ATP to convert to ADP? Because there are a lot of negative charges in these phosphate groups, and there's a lot of repulsion. So so the molecule becomes more stable if it is in the ADP form, the diphosphate form, because there are less bonds. Okay, so converting ATP to ADP is favorable because of that. Okay, so the idea here, whenever we have this reaction right here, ATP plus H2O giving us ADP plus inorganic phosphate, the delta G naught prime of this reaction is actually minus 7.3 kilocalories per mole, meaning that the reaction is exergonic. It is a favorable reaction. Now, in cells, ATP is not really hydrolyzed directly, and the energy is not released as heat as a result of ATP hydrolysis. Rather, this energy is transferred into another reaction that requires energy. Okay? So what they do is that they form an intermediate molecule that is phosphorylated, that has that would have a high energy bond. So whenever you have this reaction right here, glucose plus ATP giving us glucose 6-phosphate plus ADP, within this reaction right here, you have the formation of a transition state. You have the formation of a molecule. Okay, you have this covalent bond, an intermediate, a covalent bond between the AMP molecule, or sorry, the ATP molecule with the glucose molecule. Okay, and then you have rearrangement of bonds resulting in release of phosphate from ATP. It becomes ADP to glucose, and glucose becomes glucose 6-phosphate. So forming this transition state is actually, it becomes favorable. So if we, for example, add a phosphate to glucose directly to form glucose 6-phosphate, the reaction is inorganic. It's plus 3.3 kilocalories per mole. But if we couple this reaction to the ATP hydrolysis reaction, the reaction becomes glucose plus ATP giving us glucose 6-phosphate plus ADP. Now delta G would be delta G of the first reaction plus delta G of the second reaction, which is in exergonic in the negative form, less than 0, and the total becomes of delta G naught prime minus 4.0. So we convert an endergonic reaction, which is the first one right here, into an exergonic reaction, a favorable reaction, by coupling it to by coupling these two reactions to each other. Okay, by coupling the first reaction to the ATP hydrolysis reaction. So this is strategy number one. Strategy number two, using activated intermediates. So in addition to having a high energy phosphorylated molecule, we can use other phosphorylated molecules, or sorry, other high energy intermediates or activated intermediates. So let's look at this reaction. Okay, that is the formation of the addition of glucose to glycogen. Now, in the first reaction, uh, we talked about this reaction. So it is an exergonic reaction because we couple the phosphorylation of glucose to the hydrolysis of ATP. So this is an exergonic reaction. Then we have an isomerization reaction converting glucose 6-phosphate to glucose 1-phosphate, and we said that this reaction is slightly inorganic. Okay, now, then we have the formation of an activated intermediate, this one by here, UDP glucose. Now, this reaction is catalyzed by an enzyme known as glucose phosphorylase. Now, what this enzyme does is that it uses the high energy that exists in UTP, which is similar to ATP, except that instead of adenine, there is uridine. So there is a lot of energy in UTP, and this energy in UTP is used to attach UDP to the glucose molecule, replacing the phosphate. Now, this is an activated intermediate. Now, in addition, you have the release of what is known as pyrophosphate. And this pyrophosphate is immediately hydrolyzed to two separate inorganic phosphates by another enzyme known as a pyrophosphatase. Okay, now look at this. Now this reaction right here is exergonic, and it can proceed further. It can proceed quickly by coupling it to two reactions. The first one is this one right here, okay? And the second one is the hydrolysis of the pyrophosphate. This is an exergonic reaction right here. So it's a favorable reaction. So what happens, and this is an endergonic reaction, right? Okay, so how can we have this reaction move forward? Quite simply, by reducing the concentration of the pyrophosphate. So this is a favorable reaction. So once you have the formation of this pyrophosphate, it gets hydrolyzed. So the concentration of this product is reduced, and that reduces the delta G of the reaction. Remember that equation that I told you about? Okay, so this reaction moves forward, and it's sort of like it pulls the other reaction along with it. Okay, and this reaction is also exergonic, by the way, so so it moves really quickly. Okay, and then you have the final reaction that utilizes the energy within the UDP glucose molecule to catalyze the attachment of a glucose molecule to an existing glycogen. Now, by the way, in biochemistry books, whenever you see this sign right here, so it cleaves a phosphate bond, whenever you see this sign right here, it means that this is a high energy bond. Okay? Now, and something else about ATP, UTP, CTP, and GTP is that ATP is used in general for reactions and other biochemical and cellular work that take place inside cells. UTP is used for combining sugars for sugar metabolism. CTP is used in lipid synthesis, and GTP is used in protein synthesis. So if you remember in molecular biology course, we talked about the importance of GTP in protein synthesis in ribosomes. If you remember that, the initiation factors and the elongation factors that are involved in protein synthesis. Now, there are other high energy compounds. So in addition to ATP, CTP, UTP, and so on, there are other molecules. And these molecules include molecules that are phosphorylated. They have this high energy phosphate bond. So here's a list of these molecules. Now, look at this molecule right here, ATP. This molecule exists like it's intermediate. Okay, it has the amount of energy that is released is actually intermediate compared to the other molecules. Now, the phosphoenol pyruvate molecule contains a lot of energy. And in fact, this is the very last reaction in glycolysis. Okay, so the energy in phosphoenol pyruvate is used to synthesize ATP molecule. Now, the 1,3-bisphosphoglycerate is also another high energy molecule, and it is used to synthesize ATP in glycolysis as well. Strategy number 3, manipulating product substrate ratio. We talked about this before, and we said that in this reaction right here, we can drive the reaction by reducing the concentration of the products, that is reducing the concentration of the glucose 1-phosphate by linking it to an exergonic reaction. Okay, so this is an endergonic reaction. So once you have the formation of glucose 1-phosphate, it gets converted immediately to UDP glucose via an exergonic reaction, and it can be driven even further by hydrolyzing pyrophosphate in another exergonic reaction. So that reduces the concentration of the glucose 1-phosphate. Okay, and that would convert this endergonic reaction into an exergonic reaction. So the reaction, as we said before, has a small positive delta G naught prime for converting glucose 6-phosphate to glucose 1-phosphate. Now the ratio at equilibrium, the ratio of glucose 1-phosphate to glucose 6-phosphate is about 6:94. Now, whenever we use the glucose 1-phosphate in this reaction, the ratio becomes 3:94. That is 3 molecules of glucose 1-phosphate to 94 molecules of glucose 6-phosphate. So you reduce the amount of glucose 1-phosphate from 6 to 3. And that makes delta G exergonic. It becomes thermodynamically favorable. So, can you calculate delta G of the reaction G6P to G1P under the new ratio? Okay, under this ratio right here. Can you calculate delta G? Now, you can do the calculation and you can refer to this website as well. Now, the fourth strategy that is related to energy is that cells use to drive reactions forward is the use of electron carriers. Now, the thing is that in the body, whenever cells need energy, what they do is that they catabolize molecules. They break down large molecules, and including glucose, for example, it's used as a fuel for generating energy and producing ATP. Now, this energy can be produced by transferring electrons, by oxidizing molecules, by just like glucose, taking electrons from glucose and fatty acids as well, to electron carriers. These electron carriers include NAD and FAD. Now, these carriers can then transfer electrons to oxygen, okay? And as they do that, they create what is known as the electrochemical gradient across the inner mitochondrial membrane, okay? Now, and then what happens is that these electrons, this gradient is eliminated by an ATPase molecule transporter. As these protons move through the ATPase, ATP is produced. So there's gradual accumulation of energy across the inner mitochondrial membrane, and creating a lot of energy potential. And then this energy potential is converted into biochemical energy stored in the ATP molecule. So remember that whenever there is an oxidation reaction, there's also a reduction reaction. So every molecule has a reduction potential, meaning that the willingness to accept electrons. Okay, so and it is known as the E naught prime. Just like having G, there is also an E naught prime, which is basically it's measured in volts. It is the energy change when when a compound accepts electrons. That is, it becomes reduced. Now, this is the reduction potential. Okay, so it is the willingness of a compound to accept electrons. So each oxidation reduction reaction has a fixed amount of energy, and this is proportional, directly proportional to delta E naught prime. Okay, so delta E naught prime is the difference in reduction potentials of the oxidation reduction pair of any reaction. And the relationship between the two is the following: delta G naught prime is equal to minus N times F times the change in the reduction potential of that reaction. Now, N is the number of electrons that are transferred. F is a constant, Faraday constant, that equals 96.5 kilojoules per volt, or 23.06 kilocalories per volt. You don't have to memorize this. If I bring you a problem, I'll give it to you in the exam. E is the reduction potential in volts, and G is the free energy in kilocalories or kilojoules. So every compound has its own reduction potential. Okay, so the ability or the willingness to accept electrons. Oxygen is the best electron acceptor. It is a strong oxidizing agent. That is, it takes electrons from other molecules. Okay, and it has a reduction potential of that equals 0.816 volts. Okay, now other molecules have their own reduction potential. Now, if reduction potential is more than 1, it means that it is an oxidizing agent. It accepts electrons. If the molecule has a reduction potential that is less than 0, it means that it tends to be oxidized. It tends to donate electrons more than it tends to accept electrons. Now, remember that the opposite reaction, the reverse reaction, has a reduction potential as well, but you have to, it's the same value, but you have to change the sign. So, for example, acetate to acetaldehyde has a reduction potential of minus 0.468. Now, if you flip this reaction, the reverse reaction, that is acetaldehyde releasing electrons, donating electrons, now the reduction potential would be positive 0.468. So the transfer of electrons from compounds to oxygen is energetically favorable. Okay, and that is associated with energy release. So electrons move from compounds with lower reduction potential, that is more negative, to compounds with higher reduction potential, that is more positive. Okay, so electrons move from these molecules to these molecules right here. Okay, So looking at an example of these reactions is the reduction of NADH and FADH2. These are the electron carriers. Now, if you look at these reactions, NADH plus oxygen transferring electrons to oxygen results in the formation of the oxidized form of this electron carrier, which is NAD plus H2O. Okay. Now, if you look at this reaction and look at the reduction potential now, the release of electrons from NADH has a change of reduction potential that equals plus 0.32. So we're looking at we're looking at the opposite, the reverse of this reaction. Okay, so this is minus 0.32. So we're looking at the reverse reaction. So you change the sign, but you keep the value. Now, if you look at the reduction potential of oxygen, it is plus 0.82. Now, again, just like delta G, these are additives. Okay, so when you add them to each other, that results in a delta G that equals about 53 kilocalories per mole, and that is a lot of energy. Okay. Now, if you look at FADH2, same thing. FADH2 donating electrons, releasing electrons, has a reduction potential, a change of reduction potential of plus 0.20. Again, so it's this value right here. And reduction, the acceptance of electrons by oxygen has a delta E naught or prime that equals plus 0.82. So again, you add these to each other and apply the sum of these two to the equation that I just showed you in the previous slide, and that gives you a delta G that equals minus 41 kilocalories per mole, and that is again a lot of energy, and that is why these electrons are not transferred directly, as you will see later on. These electrons are not transferred directly from NADH or FADH2 to oxygen. Rather, you have buildup of the electrochemical gradient across the inner membrane of the mitochondria, so that this energy that accumulates across the inner membrane of mitochondria can be converted into biochemical energy in ATP molecules. Now, lastly, we want to talk about the concept of thermogenesis, that is the formation of heat or the generation of heat. Now, remember, in the first law of thermodynamics tells us that no energy is lost, so it can be converted. Now, it can be converted into kinetic energy, like, for example, opening and closing transporters on the plasma membrane. It can be converted to biochemical energy, okay, formation of ATP, and it can be converted to energy, to heat, for example. So thermogenesis is basically the production of heat as a consequence of oxidizing fuels. So whenever you have oxidation of molecules like glucose, for example, breaking up these large molecules like glycogen, fatty acids, and so on, you have the production of energy. But at the same time, you can have the production of ATP as well. So whenever we feel cold, in the winter, for example, we have this reaction of shivering, right? So we go like something like this, right? So the idea behind it, this is a natural response of our body that results in the production of heat in addition to some ATP. So this movement of muscles, this muscle contraction results in the production of ATP as well as some heat that warms us up. Now, we have also non-shivering thermogenesis, which is generation of heat without the production of ATP, and this is a not very efficient way, and it results in the oxidation of more fuel in order to produce ATP.